Good morning, folks.
Good morning.
How's everybody doing today? Okay, well, welcome back. And thanks for coming out. Traditionally, a day or two before the holidays is challenging for attendance, but I understand. I hope you're making plans to have a nice Thanksgiving. If you're traveling, please be safe. Okay, so we're just getting a few more people logged in here and we'll get started. Okay, so folks, let me start with some information here before we get into the new conversation of today's lecture. And so first thing I'll mention to you is the final exam schedule has finally come out. And so I can tell you when your final exam for this course is going to be. I'm going to type it into the chat window for you. Okay, so hold on a second. And here it comes. Okay, final exam for this course, for your course, is Wednesday, December 15th at 8 a.m. Okay, Wednesday, December 15th at 8 a.m. is your final exam. So please take note of that. And if you're in contact with classmates, please be sure to spread the news to them. I will continue to remind everybody, obviously, in the remaining weeks that we have, right? You have 6 lectures left, including today, right? Things are wrapping up very quickly here. So try your best to stay energized and have a strong finish to the semester. I know it's hard. Everybody's getting tired. Okay, so do you have any questions about the final exam? Okay, all right. Well, let me know. We still have a few weeks, so there'll be plenty of time to ask questions about it. Once again, your final exam, December 15th at 8 a.m. You will have 2 hours of time to take that exam, okay? All right. So also this morning, I wanted to share with you an interesting and kind of exciting finding in the research arena here. Hold on a second, I have to let a few more students into the class. Okay. All right, so I just wanted to share a recent result of some research up at the Brigham and Women's Hospital in Massachusetts. Okay, so let me show you this. I'm going to share my screen. There we go. Take a look at this headline, folks. This headline says, cancer cells use tiny tentacles to suck mitochondria out of immune cells. Okay, so I'm not going to go into a long-winded conversation, but I'm sure we all know that cancer is a very bad disease. And so what researchers have discovered up in Massachusetts is that in this case, they were using breast cancer cells. These cancer cells here are extending a tiny little tube called a nanotube, and they're puncturing through the membrane of a T cell. T cells are T lymphocytes. They are part of our immune system. They're part of the defenses we have to protect ourselves from bacteria, viruses, and cancer. And it turns out that researchers have discovered that cancer cells are pushing these little tubes through the T cell and literally sucking out, pulling out the mitochondria of these T cells. So remember, the mitochondria are the cell's power plants. They're responsible for manufacturing ATP to provide all kinds of energy, right? And so the cancer cell seems to be pulling out the mitochondria, which leaves the T cell in a weakened state because you're pulling out its power supply, and the mitochondria are going into the cancer cell and helping energize the cancer cell, give the cancer cell even more energy. I mean, this is an amazing finding. Who would have thought that a cancer cell can literally pull mitochondria out of another cell? We've never seen this before. I mean, this is this is really an amazing finding. So hopefully with that new piece of information, we might be able to develop some new strategies to combat cancer, because I'm sure we all know folks that have passed away due to cancer. Okay, so every once in a while, some amazing things come out of the research lab. Okay, any thoughts or questions about this? Okay, so I just wanted to share that with you. There's that old phrase in life that truth is stranger than fiction. Truth is stranger than fiction. Well, when you get into research, that is often the case. Nobody ever thought that a cancer cell would do this kind of thing. Okay, so folks, I'm going to pause the recording because what I want to do. Okay, back to chapter 12. Let me share my screen and we will get started here. Okay. All right. I will be showing you a couple of videos today on the nervous system. And so let me remind you of videos you have access to in Wiley Plus. Okay, so I'm in Wiley Plus. I'm going to go to Wiley Course Resources. Within Wiley Course Resources, I'm going to go to the Course-Wide Resources tab, right there at the top. Then I'm going to go down to 3D Physiology and open that up. There are two videos that are important for Chapter 12. They are this one, Membrane Potentials, Graded and Action Potentials. And this one over here on the top right, Membrane Potentials, Excitable Cells, and the Resting Membrane Potential. Both of these, I may not get both of them shown today, but you should watch them on your own, and I will also try to show them during class, either today or after Thanksgiving break, okay? So please be aware of these videos, which are very helpful for understanding the content of Chapter 12. All right, gang, so last time we were talking about neuroglial cells, and we were looking at figure 12.6. Figure 12.6, neuroglia of the central nervous system. In your notes last time, I put information about astrocytes, oligodendrocytes, microglia, and ependymal cells. That's where we finished. So I'm not going to continue with that. Okay. What I'm going to do is turn the page and talk to you about neuroglia of the peripheral nervous system. Relevant figures right now are figures 12.7 and 12.8. Okay, so let's get started here. The first category of glial cell that's very important for peripheral nervous system functioning are the Schwann cells. And this would be, again, I'm going to put in up here, I'll reference the figures 12.7 and 12.8. Okay. All right. So let's have a look at what we can say about Schwann cells. Okay. Schwann cells, you can see them, for example, figure 12.7, panel A or B there, really A, B, or C. If you look carefully in figure 12.7, you will identify the Schwann cell. Okay. So Schwann cells form the myelin sheath in the peripheral nervous system. And you may recall from our conversation last time that the central nervous system also has myelin sheath. So sometimes I think students have heard in other courses that the peripheral nervous system has myelin sheath and the central nervous system does not. That is not correct. Both the central and peripheral nervous systems contain myelin sheath, but the myelin sheath is a bit different between the central nervous system and the peripheral nervous system. So the Schwann cells make up the myelin sheath in the peripheral nervous system. Schwann cells are analogous. They are comparable to the oligodendrocytes of the central nervous system, which form the myelin sheath in the central nervous system. Okay, so just give me a moment, and we we will talk about what myelin sheath is and why it's important. Okay, so Schwann cells are a big deal. They form the myelin sheath in the peripheral nervous system. The Schwann cells have the ability to help regenerate damaged sections of axons. So for example, if you had nerve fibers in your peripheral nervous system that were damaged due to some kind of autoimmune disease, like multiple sclerosis or type 1 diabetes or lupus, then Schwann cells have the ability to help repair the damage. Obviously, it's never perfect, but Schwann cells can be involved in repairing damaged neurons. Okay. All right. So let's talk more about myelin sheath here. Myelin sheath. Okay. What is it and why is it important? Okay. So, all right. So a couple talking points here. Okay. And figure 12.8 does a nice job of explaining what myelin sheath is. Okay, myelin sheath is a wrapping of a Schwann cell in the peripheral nervous system or an oligodendrocyte in the central nervous system around an axon multiple times. Okay, so myelin sheath is a multi-layered wrapping of Schwann cells in the PNS or oligodendrocytes in the CNS around the axon of a neuron. Okay, so there's a subtle distinction here. If I were to use an analogy, imagine for a second that your finger is an axon. Imagine that your finger is an axon and that you get a small paper cut on your finger. If you get a small paper cut on your finger, you probably are going to put a Band-Aid on it. So the Band-Aid is going to be our Schwann cell. If you had a Band-Aid and you put it on your finger and you wrapped the Band-Aid around your finger one time without the Band-Aid wrapping on top of itself, that is technically not myelin sheath. If you have the Band-Aid go around your finger once with no overlapping of the Band-Aid on top of itself, that is not myelin sheath. Myelin sheath is when you have the Band-Aid go around your finger and then you continue to wrap the Band-Aid on top of itself multiple times. So you build up a thick wrapping of Band-Aid around your finger. That is myelin sheath. So analogy here, one layer of Band-Aid around your finger is not myelin sheath. Multiple layers of Band-Aid around your finger is myelin sheath. Question in the chat window for you. Oh, I can't type well today. Here we go. Are you okay with the subtle distinction of what makes up myelin sheath? One layer of Band-Aid, not myelin sheath. Multiple layers of Band-Aid wrapped on top of itself several times, that is myelin sheath, okay? All right, so that's what the myelin sheath is. Let's talk about why it is important. Okay. So let's list this down as functions of myelin sheath. Okay. There's three things that I'm going to say here. First off, myelin sheath acts as an electrical insulating tape. Acts as electrical insulating tape to prevent electrical signals from jumping to neighboring axons. Okay, now that in and of itself is kind of a peculiar idea, so let's talk about that for just a brief moment. So I'm going to just draw a little cartoon here, and hopefully you'll get the main idea. Okay, so if my technology will work here. Okay, so let's say that we have all these axons that are running next to each other inside a nerve fiber, right? Because remember, we defined that a nerve is a parallel bundle of axons from hundreds to thousands of neurons that are held together with connective tissue and nourished by the blood supply. So here we have multiple axons, and realistically, these multiple axons are encased inside the connective tissue that would be a nerve. So this whole thing is a nerve, one nerve with multiple axons inside it. And so you appreciate that, you know, a given axon might be propagating an electric signal. So I'll just indicate this by these green little arrowheads. So we have an electrical signal going down here. Because the axons are so physically close to each other, the distance between the axons is very tiny. I couldn't give you a number off the top of my head without looking it up in some, book somewhere, but I suspect the distance between the axons is probably no greater than 50 to 100 nanometers, if that. Okay, and so the danger here is when you have these axons so close to each other, what can unfortunately sometimes happen is that the electrical signal actually can jump over to the neighboring axon. That would be a short circuiting, and you don't want that to happen, because that means that the electrical signal traveling down the top neuron in our drawing is going to get to the wrong destination because it did not continue its journey along its intended path. So that's a bad thing. We don't want that to happen. Myelin sheath can prevent that from happening, because myelin sheath, and please forgive me for not being terribly artistic here, myelin sheath, if I do this, myelin sheath wraps around the axons, and I'm just going to indicate myelin sheath with these kind of little orange rectangles. These orange rectangles would be multi-layered wrappings of the Schwann cell's plasma membrane around the circumference of the axonal fiber. Okay, so this would be, each one of those little rectangles is a Schwann cell. And all the Schwann cells together as a group of Schwann cells would be the myelin sheath. Okay. And so what I'm telling you is that the presence of the myelin sheath prevents the electric signal from a neighboring axon from jumping across. It prevents that because it insulates the axon inside the myelin sheath and it prevents that from taking place. Okay, so that's what I mean by point Roman numeral little I there. Myelin sheath acts as electrical insulating tape to prevent electrical signals from jumping to neighboring axons. Any questions, folks? Okay, all right. So that's the first thing. The second thing of myelin sheath is as follows, and this one's going to be weird too. Believe it or not, the presence of myelin sheath has the strange ability to accelerate the transmission speed of the electrical signal. The presence of myelin sheath makes the electrical signal travel the same distance faster than if the axon had no myelin. Okay, myelin sheath accelerates the transmission of the electrical signal. This is a phenomenon called saltatory conduction, a phenomenon known as saltatory conduction. And so what I will say is, I'm not going to spend too much time on this, and I would encourage you to read up on saltatory conduction, maybe even do a YouTube search for saltatory conduction, watch a video or two about it. But the basic idea of saltatory conduction can be described in an analogy, okay? And so I'll do this, okay? And I'm going to use basketball as my analogy. So I hope all of you at least know what basketball is, even if you don't play it or enjoy watching it. Honestly, it's not my favorite sport either, but hopefully you know what a basketball is and you've seen a basketball court, okay? And you've seen basketball nets, right? A lot of playgrounds have basketball nets. Children like to play basketball. So here's the analogy. If you're playing basketball and there's a basketball hoop on the other end of the court and you have the ball, there are three potential ways that you could make a shot and get it in the net, right? Number one, wherever you're standing, you could do a Hail Mary toss and just throw the ball as hard as you can and as far as you can and hope it gets in. A lot of times you'll see that kind of shot at the end of a basketball game when the clock's running out and they just throw it and hope something goes in, right? So that's the first possibility in basketball, and I'm here to tell you that that never happens in the nervous system. That does not happen in the nervous system. So just throw that out. The Hail Mary pass does not exist. The second thing that you could do in basketball, if you have the ball and the hoop's at the other end of the court, you could take and you could literally dribble the ball down the whole court yourself until you're next to the hoop and then take your shot, right? That, folks, is how the electrical signal spreads down a neuron in an unmyelinated neuron, an unmyelinated axon, okay? Dribbling the basketball down the entire court. This is comparable to moving the electrical signal down an unmyelinated axon, okay? Continuing with our basketball analogy. If you have myelin sheaths around an axon, then this intersects with the following idea from basketball, okay? So we just said dribbling the ball down the court is similar to the transmission of an electrical signal in an unmyelinated axon. There is a third possibility that happens in basketball. If you have the ball, you could take it and you could pass it to one of your teammates who's already standing closer to the net. So you could pass the basketball. Well, passing the basketball is similar to passing the electrical signal along the axon. That is saltatory conduction. Passing the basketball is saltatory conduction. Passing the basketball is passing the electrical signal, and that only takes place along axons that are wrapped in myelin sheaths. Okay, so passing the basketball to a teammate closer to the hoop is similar to moving the electrical signal down a myelinated axon. Okay, so without wanting to spend really too much more time on this, what I'm saying to you is, if I zoom in here with the drawing we have, okay, and I will use, let's see, I'll use yellow to indicate kind of the spark of an electric signal. In an unmyelinated axon, such as we have at the top, you're going to have the spark of the electrical signal as a dribbling ball down the basketball court. Here's the spark of the electric signal. I'm dribbling it the whole way down the court. You can see how tedious it is to move the electrical signal down the court. Each one of these X's is a dribbling of the basketball. Each one of these X's is an electrical signal event known as an action potential. Okay. In a myelinated neuron, I don't have to dribble the ball as much. So I can have an electrical signal here, and here between the Schwann cells, and here between the Schwann cells, and here between the Schwann cells, and here between the Schwann cells. So I actually can move the same distance faster by triggering fewer electrical signals known as action potentials. Okay. If that's not entirely clear at this time, hopefully it will be clear by the time we get some videos watched during today's class, okay? So, dribbling the basketball equals the X, which is the same thing as an action potential. Okay, folks, do you have any questions you'd like to ask? I know this is not the most intuitive topic. Understanding how the nervous system works, you know, I mean, let's be honest, you've had to deal with many complicated topics in this course, and this is just another one on top of the Sunday. But this is a pretty meaty, difficult topic to understand. It will require some really dedicated time on your part. Any questions? Folks, videos are going to help a lot, okay? So I'm going to show some videos, and I want to encourage you to use videos during your private study time. Okay, so I'm going to shrink that back down so I can take some notes here. Okay, and we'll continue. All right, so this is all talking about the myelin sheath, right? That's the whole purpose of this conversation. We're talking about the myelin sheath. We've defined what it is. It is a multi-layered wrapping of Schwann cells in the peripheral nervous system, or a multi-layered wrapping of oligodendrocytes in the central nervous system. That's great. Well, what does it do for us? It acts as an electrical insulating tape to prevent short circuiting. It also accelerates the transmission speed of the electrical signal. The last and probably most boring thing I could tell you is point number 3, is that since Schwann cells and oligodendrocytes are types of glial cells, it should not be a surprise to you that one of the functions of myelin sheath is to physically protect and nourish the axons of the neurons that they wrap around. Okay, so to protect and nourish neurons. Okay, folks, do you have any questions right now? All right, let's continue. All right, so we were talking about Schwann cells as an example of a neuroglial cell of the peripheral nervous system. There's one other category of glial cell in the peripheral nervous system that I'm not going to spend a lot of time on, but I am going to put a few notes into this document for you, okay? That would be back to figure 12.7. And it would be the satellite cells. Take a look at figure 12.7, picture C as in Charlie. Picture C as in Charlie. Satellite cells are another type of glial cell. Satellite cells surround the cell bodies of neurons that are part of ganglia in the peripheral nervous system. And so from that perspective, satellite cells provide sort of protection and support to the neurons that are part of the ganglia. Surround and protect / support neurons that form groups in the PNS called ganglia. Okay, so let me ask you a question in the chat window. What are groups of neuronal cell bodies in the central nervous system called? Okay, all right, thank you, Shay. Good job, you're absolutely right. Within the central nervous system, if you have a group of organized cell bodies, it's called a nucleus. But in the peripheral nervous system, the analogous structure is going to be a ganglion. All right, so satellite cells surround and protect the cells of the ganglion. That's one of their functions. The second function is to regulate the exchange of material, to act as a go-between, to act as a bouncer at a nightclub for the exchange of material between the cell bodies that are part of the ganglion and the surrounding interstitial fluid, which you know is formed from the bloodstream. So basically, the satellite cells act as a protective filter to make sure that dangerous toxins don't get to the neurons. Okay. All right, so regulate the exchange of materials between the cell bodies of cells in the ganglion and the surrounding interstitial fluid. All right, folks, so if we think about it, we put into your notes last time four different types of neuroglial cells in the central nervous system, and now we have placed 2 different types of neuroglial cells in the peripheral nervous system into your notes. Do you have any questions before we move to the next topic? Okay, the next topic is, drum roll, the electrical signal. Now we need to really start talking about this beast, and we're going to be talking about it the remainder of today and all of next lecture on Wednesday. Okay, so let's do this. Electrical signals. And we're going to start with some background or overview information. Okay. Within your nervous system, and it really doesn't matter whether I'm talking the central or peripheral nervous system. Within the nervous system, the whole shebang, there are basically 2 different types of electrical signals. Many folks out there in the universe have heard of action potentials. That's only one of the two types of electrical signals. The other type, which is much less famous and lesser known, is known as the graded potential, okay? So there are two types of electrical signals in the nervous system. We'll be discussing both of them with our time together. Okay, the two types are graded potentials. Graded potentials are used for very short distance communication between cells. Okay, very short distance communication, and I'll qualify that in just a moment. Used for short distance communication. In this context, short would be defined as a few millimeters, Short would be a few millimeters, 3, 4, 5 millimeters. Okay, so, you know, if you happen to have a ruler at your house, right? A lot of rulers have the standard King's English system on one side with inches, and on the other side, you've got the metric system with centimeters, right? Centimeters, and in between the centimeters are even smaller divisions called millimeters. Well, I would say that graded potentials typically are measured in a few millimeters, okay? So graded potential is used for very short distance communication between cells. Action potentials are used for much longer distance communication. And then in this context, long could be upwards of a meter, right? A meter is 39 inches. If you think about it, we have some very tall people in society, right? Think about your basketball players, right? That can easily be 6'6" or taller, right? And so, and a neuron, the axon of a neuron could run down from that person's hip the whole way down that long, thin leg of the basketball player to their foot. That could easily be 3 feet or longer in length. And it's that kind of distance we're talking about for an action potential that could propagate along our anatomy. So long is defined as 1 or more meters. Okay, 1 or more meters. Remember, a meter is 39 inches. Okay, so graded potential, you know, for a graded potential, imagine a dime, a 10-cent coin that you might use at the grocery store to buy some food, a dime. Think about the diameter across a dime. That would typically be the distance of a graded potential. Think about the length along somebody's leg, even if they're 7 feet tall. That would be the distance of an action potential. Okay, number 2 here. There's different terms that are basically used to describe the electrical signal. Okay, so the electrical signals, electrical signals are also called impulses. Okay, electrical signals are also called impulses. And the nature of the electrical signal or impulse is contextual. And what I mean by contextual is it is described from the perspective of the type of cell that is generating the impulse or generating the electrical signal. Okay, so when an electrical signal is created inside a neuron, we call this signal a nerve impulse. Okay, we call this signal a nerve impulse. When an electrical signal is created inside a muscle fiber, we call this signal a muscle impulse. Okay, so, I'm here to tell you that an electrical signal is an electrical impulse. And if the impulse is generated in nervous tissue, we call it a nerve impulse. If the impulse is created in a muscle fiber and muscle tissue, we call it a muscle impulse. Okay? And it is also true that the nerve and muscle impulses are basically the same. Are there some subtle differences in the electrical characteristics of a nerve impulse and a muscle impulse? Yes, there are, particularly when we compare the electrical signal in a neuron to the electrical signal of cardiac fibers in your heart. There are some subtle differences, yes. For right now, we won't focus on the differences. We'll focus on the commonalities so you understand what the electrical signal is. But you might also hear the phrase action potential. And action potential is basically the electrical impulse, which is the electrical signal. Okay, so action potential is the same thing as the electrical signal, which is the same thing as the impulse. Okay, all of these terms are used in different books in different ways, and they're basically meaning the same thing. Okay, folks, any questions right now? Okay, let's continue. All right, so, if I use the phrase action potential or electrical signal or nerve impulse, it's all the same thing, folks. For our conversation, it's all the same thing. All right. One final piece of background information is this idea of membrane potential. Okay. Membrane potential is a phrase, it's a term, if you will, that refers to the separation, or you might alternatively say the difference in electrical charge on two sides of a membrane. The membrane that we're interested in here in biology is always going to be the plasma membrane of a cell. So the membrane potential is the difference in electrical charge between the inside and outside of the plasma membrane of a cell. Okay. This refers to the separation or difference in electrical charge between the inside and outside surfaces of the plasma membrane of a cell. Okay, the difference in electrical charge between the inside and outside surfaces of the plasma membrane of a cell. This is membrane potential. Membrane potential, the difference in electric charge, is typically measured in units of millivolts. Okay, and so if you realize that the idea of membrane potential is measured in millivolts, then it starts to make sense when you talk about things like voltage gated calcium channels that we talked about in the neuromuscular junction. Okay, this idea is measured in units of millivolts. In other words, if you had a membrane, let's say my hand is a membrane, and let's say you had two electrodes. One electrode you could put on one side of the membrane, and the other electrode you could put on the other side of the membrane. You could measure the difference in charge between the position of those two electrodes on the opposite ends of the membrane. That would be membrane potential. Okay. All right, so this is all kind of good, but yet not kind of good. It's kind of like we're defining things that you kind of get, but then you don't really get it. You kind of grasp it, but then it slips away unless we beat it down to a pulp. So let's start beating this down to a pulp. And so I'm going to start by showing you the first video in this chapter, again from Wiley Plus. Okay, so give me just a moment to cue this video up, and don't take notes the first time, because you have access to this yourself in Wiley Plus, and I want to encourage you to watch the videos several times till you kind of start making sense of the notes we're giving you, what the textbook is telling you, what the video is showing you, so you have a triangle of support information to hopefully get this across to you. So let's watch something here. I'm going to show you the graded and action potentials video. Here we go, folks. Just sit back and enjoy.
Membrane potentials, graded and action potentials. In order to describe the generation of changes in membrane potential, we will use a motor neuron, which stimulates skeletal muscle to contract. The neuron can be divided into three zones. The input zone, consisting of the cell body and extensions known as dendrites, has chemically gated channels in its membrane to receive stimuli from other neurons as graded potentials. These graded potentials move toward the trigger zone at the axon hillock. The trigger zone is where voltage-gated channels are first encountered in the neuron. If the graded potentials reach threshold in the trigger zone, then an action potential is generated. The action potential is transmitted via voltage-gated channels down the axon to the output zone, where it causes skeletal muscle to contract. In the input zone, graded potentials are generated when stimuli such as neurotransmitters attach to receptors of chemically gated ion channels, causing them to open briefly. Then, shift in the distribution of ions on either side of the membrane results in a slight change in electrical potential. Graded potentials travel a short distance through an input zone toward the trigger zone before fading away. It is the net voltage produced by graded potentials at the trigger zone that determines if the threshold of voltage-gated channels is reached to trigger an action potential. Some graded potentials occur when chemically gated sodium channels open to allow sodium ions to diffuse into the input zone. This electrically moves the trigger zone closer to or above the threshold. They are considered to be depolarizing graded potentials. If chemically gated potassium channels in the input zone are stimulated to open, it allows potassium ions to diffuse out of the cell and causes the inside of the membrane to become more negative, moving the voltage in the trigger zone away from the threshold. These inhibiting graded potentials are known as hyperpolarizing graded potentials. Since depolarizing graded potentials cannot individually reach threshold, an insufficient quantity does not cause a response at the trigger zone. If, on the other hand, there are enough of them to reach threshold, an action potential is generated. If there are more hyperpolarizing graded potentials than depolarizing, the net membrane potential in the trigger zone becomes even more negative than the resting potential. This would temporarily inhibit the generation of an action potential or require additional positive stimulation to reach threshold. When the net graded potentials arriving at the axon hillock reach threshold, the action potential begins. Both sodium and potassium voltage-gated channels are stimulated to open. Sodium channels open quickly, allowing sodium ions to diffuse in and add positive charges to the inside of the cell membrane. If enough positively charged sodium ions diffuse in to bring the voltage on the inside of the membrane to 0 millivolts, it becomes uncharged, and we refer to the cell membrane as being depolarized. Sodium channels are only open long enough to allow sufficient amounts of sodium ions to pass through to cause depolarization. At this point, the inactivation gate on the sodium channel closes to block any further inflow of sodium ions. In actuality, before the sodium channels close, enough sodium ions diffuse in to cause the inner surface of the cell membrane to become positively charged. The purpose of the inactivation gate is to prevent the membrane from stimulating itself and causing repeated action potentials. At the same time the sodium channels close, the slower voltage-gated potassium channels open and allow potassium ions to rapidly diffuse out of the cell. The removing of positive charges from the inside of the membrane causes its internal surface to become negatively charged again or repolarized. Once enough potassium has diffused out of the cell to cause the inside of the membrane to be below threshold, the inactivation gate of the sodium channel opens as the gates on both types of ion channels close, but not before the inside of the membrane becomes less than the resting potential of minus 70 millivolts to cause the inside to become hyperpolarized. Sodium-potassium pumps transport sodium ions out of the cell and potassium ions into the cell to restore ion concentrations and to, along with leak channels, re-establish its resting membrane potential. The depolarization of one area of the cell membrane causes neighboring voltage-gated sodium and potassium channels to reach their threshold. The result is the action potential spreads like a wave across the entire cell membrane. Once the action potential begins, all of the output zone will depolarize and repolarize. This is known as the all or none principle. In other words, the action potential does not diminish as it travels across the membrane, unlike the graded potential. So either all of the membrane depolarizes and repolarizes, or none of it will. It is also important to note that the amplitude of the action potential is always the same. It is the frequency of the action potentials that signifies a stronger response.
Okay, folks. So, hopefully,
Membrane potentials, graded.
hopefully that's just a tiny bit clearer than it was 6 minutes ago, even though it may not be completely clear yet what's going on. We're going to be talking about this the rest of today and on Wednesday, so we're not done. Do you have any questions you'd like to ask from having seen the video one time? It's a lot to take in at one moment, isn't it? It's just a lot to take in. So we need to keep wrestling with it over and over again until it finally starts to stick. Okay, so lots more to do here. Please ask questions along the way. All right, so you noticed in the video, it kept talking about these ion channels, particularly in the video, it talked about sodium and potassium ion channels. And so that's where we need to focus our energy next. Okay, so I'm just looking at the chat window. Noah, I see your statement that you're not even sure where to begin to have questions. Yes, and that's often the case, right? A topic seems so overwhelmingly complex that you don't even know where to begin. And so my advice, Noah, is start anywhere, and start picking it apart one bit at a time, okay? So let's continue here. All right, so let's talk about these ideas of ion channels, okay? You'll notice that your book talks a lot about ion channels in figure 12.11, figure 12.11, okay? So go ahead, and if you have access to your book at this time, take a look at figure 12.11. We need to talk about ion channels. We've already talked about an ion channel in this course, right? We talked about the voltage-gated calcium channel. So let me ask you in the chat window. Here we go. are the voltage-gated calcium channels of the neuromuscular junction represented by panel A, B, C, or D in figure 1211. Which one is it? Which one is it? A, B, C, or D that's showing you the essence of a voltage-gated calcium channel? Is it A, B, C, or D in figure 1211? Just take a look at the information there in the figure, folks. Don't make it harder than it has to be, okay? A, B, C, or D, which is the voltage-gated calcium channel drawing? Okay, all right. Shay, you say D as in David, and you are spot on. Good job, okay? So, folks, take a look at letter D there. That's the voltage-gated calcium channel. That's what we were trying to describe in lesser detail at the neuromuscular junction, last week or two when we were talking about this. So, okay, ion channels. These are proteins, as you know, that are made of amino acids, okay? And the amino acids come together in such a way so that the inside of the protein develops a little hole or a little channel. I can't really do it so good with my hands, all right? But here's a tube which represents a protein, and you can look through the tube, right? There's a little hole there. That's the channel. So the ion channels are made of proteins that there's a tunnel inside or a channel inside that ions can move through that tunnel and get to the other side of the membrane. They can either get into the cell or out of the cell by moving through the tunnel that's in the interior of the protein that's embedded in the membrane. These are made of proteins sticking through cell membranes. These proteins have a hollow tunnel or channel that allows ions to move into or out of the cell. Okay. Now, when you talk about the motion of ions into or out of the cell, you need to refer to something known as the electrochemical gradient. So let's define that. So hold on, before I define it, let me finish this idea of ion channels. These are made of proteins sticking through the cell membranes. These proteins have a hollow tunnel or channel that allow ions to move into or out of the cell. The ions will always move down their electrochemical gradient. That is to say, they will move to an area where there is a high electrochemical energy to a lower electrochemical energy. All right, let's define that. Electrochemical gradient. Okay. Folks, a gradient is an area of difference. An area of difference. Think about it this way for a moment. We've all been, at some moment in our life, you took a walk somewhere, whether it was through the streets of a city or out in the countryside in a park. Maybe you were fortunate enough to do some hiking in the mountains, whatever. The point is, a gradient is an area of difference. So imagine you're walking along and you start to climb up a hill. So when you're on top of the hill, that's your position. And then when you go past the hill and you start walking down the hill, you're now below where you were previously. So as you walk up the hill, you're at one point in elevation. And as you walk down the hill, you're at the second lower point of elevation. The difference in altitude between those 2 points at the top of the hill and the bottom of the hill is a gradient. The difference in elevation between the top of the hill and the bottom of the hill is a gradient. It is a difference in elevation. Now, the gradients we're talking about is not about a difference in elevation. It's about a difference in electrical energy and chemical concentration, okay? So the electrochemical gradient is a gradient, a difference, between two locations with respect, okay? Electrochemical gradient is a gradient, a difference between two locations. Those two locations are typically inside and outside a membrane with respect to two things. Number one, the electrical status of atoms in that location or between those locations, the electrical status or energy of atoms between those two locations. And two, the concentration, the number of molecules or the number of particles of atoms present in those two locations. The concentration of atoms between those two locations. Okay, let me read this to make sure I didn't mess up your notes here. An electrochemical gradient is a gradient. It is a difference between two locations, inside and outside a membrane, with respect to two things. Okay, so it's a difference in elevation if we're using our walking analogy. There's two aspects of an electrochemical gradient are the electrical status of the atoms between those two locations. That's the electro part of the electrochemical gradient. And the concentration of atoms between those two locations, that is the chemical part of the electrochemical gradient. Okay. Oh dear, I don't know if I'm going to get this finished here. So let's do an example here. I'm going to ask if you have the luxury of holding on to giving me like 3 minutes of time and staying a little bit after. That would be very helpful if you can do it. Okay, if you can, I understand. Let's do this, and I'm going to do this by drawing. Okay, let's say to help try to make this more understandable, let's do two examples here. Example one, let's say we have a membrane. Okay, so here's a membrane. Let's say on one side of the membrane, I have, okay, so I'm gonna say that this left side, I'm gonna call it side A, and this right side, I'm gonna call side B. Okay. Let's say on side A, I have 100 positively charged sodium ions, and on side B, I have 25 positively charged sodium ions. In this scenario, you're always going to have the ions on side A move towards the ions in side B. Why? Because we have the gradient, the highest part of our hill is on side A, and the lower part of our hill is on side B. If we think about the definition of an electrochemical gradient being a difference between those two locations, here's what I can do, right? I can have 100 minus 25 being equal to 75, right? That minus is the difference. That minus is the difference. That is also the membrane potential, which would be measured in millivolts. Okay, so that's example one. I'm not quite done yet, folks, but give me some feedback in the chat window. Are you with me? Is this okay so far? Okay, I know this is rough and it's going to take a little bit of time. Now let's do example 2, and I'll try to squeeze it in at the bottom rather than running it off the page. All right, so let's do the same thing with example 2. Let's say we have a membrane. We have side A. Computer. Side A, let's say we have 100 sodium ions on side A, 25 sodium ions on side B. But now, in addition to this, I'm going to put, and let's see, I'll use orange, or no, I'll use blue. Let's say on side B, I'm going to put 25 potassium ions that also have a positive charge. So the difference here in energy is now going to be, excuse me, I can't run off the bottom here. It's going to be 100 minus 50. Where did I get 50? Where I added 25 positive ions of sodium and 25 positive ions of potassium. So that now means that my difference is 50. Okay. So I'm going to ask you a question in the chat window right now. Using the hill and elevation analogy, which example, 1 or 2, has a higher hill? Good. I'm seeing the right answer. Example 1 has the higher hill. Example 1 has a larger difference. A larger difference in elevation. A larger difference in elevation. A larger difference in electrochemical gradient. A larger difference in electrical energy and chemical concentration between the membrane. So example 1 is the higher hill. Let me ask you this, and then we'll let you go. Will a ball roll down faster on a higher, more steep hill, or a lower, more shallow hill? Which kind of hill is going to allow the ball to roll down faster? Going to be the higher one, right? More steep one. So what I'm telling you is, since example 1 in your notes has the higher hill, it has the greater membrane potential, it has the larger difference. That means the ions from side A in example 1 are going to travel across the membrane more aggressively, faster than the ions in example 2 will move across the membrane. Okay, I'm done. I promise I'm done. Okay, folks, any questions? All right, we'll stop here. And folks, not much else to say. You know that Wednesday we do have lecture, but we will not have lab. So please try to log in Wednesday. We will continue this conversation, try to make it even clearer to you what all this electrical business is about, okay? Until then, take care, and I'll see you Wednesday.
Thank you, Professor. Have a good day.
Thank you.
Bye-bye.